Hello there, microbiology students. I'm here to give you your first lecture, and as you can see, it's entitled The History of Microbiology. And so the goal here is to understand some of the important figures who have contributed to what we know in the field of microbiology. So with that, we will move on to the next slide. So what you see here is a list of different individuals, and on the right are the disciplines that we recognize today that are really important in the field of microbiology. So this is not something to memorize, but just to understand why we're looking at this. So why are we trying to have this historical perspective? And so these are just two of the things that we're going to cover. You hopefully remember from your introductory biology class the importance of the building block of all living things, which we call the cell. And so there's one individual who was first responsible for describing that fundamental unit of life. And then obviously our understanding of microorganisms that we can shorten and call microbes. So that's another really fundamental part of microbiology, is the organisms that make up this field. And so we aren't going to cover every single one of the people along this list, but we're going to go through a good number of them. And so you'll see these names on future slides. And again, the goal here is just to understand that these individuals have laid the groundwork for many of the things that we're still studying to this very day. So there's a lot going on this slide, and what we want to look at is just the first individual. So his name is Robert Hooke, and I have a date here just so you know when this is occurring within the timeline of things, but I'm not asking you to memorize dates. The goal here is to understand the person, one, and what their contribution is. So those are the two things that you should focus on. And so Robert Hooke is that individual who I mentioned earlier that came up with the term cell that we still use today. And the reason why that's so important, cells are the fundamental building block of all living things. So there are some organisms that are just one cell, so a single cell. But then there are others, like humans and most animals, that are many cells, so trillions of cells that work together. But the cell theory is one concept that's really important in the field of biology, and because of that, also in the field of microbiology. So there's a video here if you want to watch it. It just gives you some more information about Robert Hooke's contribution. I'm not going to play it for you, but it's embedded within this PowerPoint, so you can feel free to watch it if that helps you better understand this. What this shows is the actual micrograph, so a picture through a microscope that he was taking. And so this is, if you look, these little units here, those are individual cells within a cork plant. And so this was him noticing that there are these building blocks. He called them cells. We still call them cells, and that's a really important finding within the field of biology. Let's stop the video. So the next individual, his name is Antonin van Leeuwenhoek, and he, unlike Robert Hooke, who was a scientist, he is not a scientist. So Leeuwenhoek actually was a garment maker, and so to get the finest quality fabrics, they really needed to investigate the threads that they were using to make different garments. And so he started to grind his own lenses, so out of glass, so that he could better visualize the threads that he was using to make different garments. So this is that little dot right there is a glass lens. And so this is what Leeuwenhoek's microscopes look like. He could put a little sample right on this needle here and then hold it up to a light source and look through it. And it's kind of like a magnifying glass, but this was a microscope. And what's really important is that he not only looked at fabric, but he looked at all sorts of things. So just like kids, when they get a microscope, whenever you enlarge something, it looks really interesting. And so he just looked at a variety of things. But what makes this the most kind of important finding is that he made very detailed descriptions. So he wrote them down and he made drawings of things. And so as he looked at things like pond water and teeth scrapings, he was the first that we know of to actually describe what we now call microorganisms. So things like little animals that you can't see with your eye, fungi, algae, even single-cell protozoans that we'll look at in a little bit. So these didn't have names, but he made up a name for them. He called them animalcules. And so that's the term he coined. We didn't keep that today because we now know there's so many things that are classified as microorganisms. And so we now use the term microbes or microorganisms, and we subdivide them into different groups. And the next lecture that you're going to look at is going to better understand those different categories. So what makes something a fungus? What makes something an algae or a protozoan or even a bacteria? Hopefully you realize they're all microorganisms, and so that's a good place to start. But Leeuwenhoek is considered the founder, one of the founders of microbiology, because it's his descriptions that we now know, being so detailed, match up perfectly with what we know what these different groups are. So included in that group is bacteria, which are the smallest of the microorganisms that you can see through a light microscope. So he saw those as well. So those are now called microbes. And again, there's a video there if you care to watch it. I'm just going to skip through it. Most of these videos are relatively short, somewhere between 2 to 5 minutes if you just want more background information on each of those important figures. So what we're moving on to here, you can see this list of questions that some of them are obviously very relevant in today's world, like what is the cause for diseases and how do we prevent infection and disease? So obviously in a world that is very much run by COVID-19 these days, these are really important questions, and they have been asked throughout history, but there's some individuals who really laid the groundwork for what we know for many of these infectious diseases, how they're actually transmitted and what causes it and how we prevent it. But prior to getting to those answers, there are some more philosophical and scientific questions that really gave the answers to those questions, gave way to understanding about disease and prevention of disease. So there is this debate about a concept called spontaneous generation that we want to look at first, and then we'll move into fermentation. But these are some of the questions that really were important that laid the foundation for a lot of what we study in microbiology. So spontaneous generation is an idea where living matter is going to spontaneously arise from nothing. And so this was looked at throughout history, and if you just watch something like a piece of bread, you see mold growing on it. There is no large organism that comes that you can see and deposit some living matter on it. It seems to appear spontaneously from nowhere. So the spoilage of food, the maggots that appear on meat, that was the evidence that people provided for spontaneous generation. We're going to look at two scientists, so Francisco Redi and Louis Pasteur, that provided evidence against spontaneous generation. Another term for spontaneous generation is abiogenesis. And so if you know kind of the roots of words, bio is life, genesis is creation, and a is without or lacking. And so this is the generation of life without living matter giving rise to it. So that's what spontaneous generation is. So the first person we're going to look at is Francisco Redi. And this experiment was really important because it's one of the early examples of using the scientific method to try to demonstrate and understand a principle. And so Francisco Redi's experiment is very simple, but it was a really important finding that pointed away from spontaneous generation. And so I gave the evidence of the appearance of maggots for why spontaneous generation could be true. And so Francisco Redi set up this experiment where he had different vessels. These are glass vessels that had meat in each. But then he allowed different sorts of access to the meat by either using cork or mesh. And we all know that when you have any food out on a picnic, flies will come and be interested in that. And the reason for that is they have to lay their eggs in some food source, and meat would provide a good food source. And so if you leave your meat just open, you can see flies interact with it. What you can see is that they can deposit eggs on the surface, and then those eggs over time will develop. They'll hatch and develop into small larvae that we call maggots. And if you continue to watch that, those maggots would further develop through a process of metamorphosis into these adult flies. Excuse me. So he noticed that, and he said, well, what if you keep the flies from interacting? So if you put a cork on that, obviously no flies can get to it, no maggots appear. You can also put a mesh screen so that those chemical odors that the flies can detect, so they try to gain access to it, but they can't actually physically interact with the meat, but they're trying to get close. And so you can actually have them depositing eggs on the surface here and larvae developing. And so Francisco Redi, his experiment very clearly demonstrated that animals, to generate more animals, you have to have a living source. So animals give rise to other animals. And so this was a really important finding, but there still was this category that we didn't fully understand of microorganisms. And so could microorganisms just spontaneously generate? Since we can't see them, it's really hard to measure that. And so while people started to accept these findings that spontaneous generation didn't work for animals, a lot of people weren't convinced in the microbe world. And so that's where we encounter the next individual, and his name is one of the kind of founding father of microbiology. You'll see his name come up over and over again. So his name is Louis Pasteur, and he used pasteurization. So he came up with the process of heating things to prevent microorganisms from growing in it. And so upon coming up with this process, he also figured out, well, maybe he could study this process of how microbes actually come to be. And so his process of heating, so here you can see a heat source, and this is just like a beef broth. So it's a nutrient-rich liquid that if you left it out just on your counter, if you opened a can of broth and let it sit there, as you might guess, it's going to start getting cloudy because stuff will grow in it. And so if you heat it, you can increase the lifespan of it. But he also coupled that with this really ingenious apparatus. And so this is called a swan neck flask. And so this is like a glass straw that vapors can move in and out of. So he heated this up basically to kill off all the existing microorganisms. Air and things in the air can move through this, but microorganisms, I'll move down this bottom one, cannot move themselves up this glass swan neck flask and then actually get to that nutrient-rich broth. They accumulate at the very bottom of this. So what this is showing is if you heat up and kill off all the microorganisms and allow it to sit, this broth will not become cloudy. And so there's no evidence that life can just spontaneously generate inside this beef broth. Instead, it appears there are these things that you can't see in the air. We now know they're microorganisms that are required to actually be the seeds that give rise to life. And so life has to be present to give rise to other life. And you can demonstrate this by taking this beef broth and swirling it around so it sloshes up into this area where there's those microorganisms. And then once they have come in contact with that beef broth and they have those nutrients, they can grow and you can see that cloudiness develop. And so Louis Pasteur was really the first person to fully demonstrate that there are these little things in the air, we now call microorganisms, that are necessary to lead to that cloudiness that develops. And once that was established, then you can start asking questions like, well, what if those microorganisms were to enter, right, our bodies? So we can't see them. And what if they get in that open wound? What if you inhale them? And so on and so forth. And so that allowed people to start answering the questions about disease and how disease is caused with the idea that there are these things called microorganisms. Before we get there, continue with Pasteur, there's another kind of offshoot of spontaneous generation. And so this next slide is talking about the process of fermentation, which has been carried out for hundreds, probably thousands of years, in terms of making wine and also making beer. And so fermentation was very much in the same realm as spontaneous generation. People would crush grapes and they just assumed that it spontaneously converted into wine. And sometimes they'd get good wine, sometimes they get contaminated wine that they couldn't sell. So it wasn't a very scientific process. So having spoiled wine was not super helpful if you're trying to make a living on it because you can't sell the spoiled wine. So again, this is very much related to, is it spontaneously occurring, or are there these microorganisms that can drive this process of fermentation? And of course, Louis Pasteur was very much in the field of, there are these microorganisms that give rise to it. So he can do a very similar experiment, but this time it has grape juice. And if you boil the grape juice, killing off all the microorganisms, and then ask the question, can grape juice spontaneously turn into wine? Well, the answer is no. You can repeat this in this swan neck flask, and you can then say, is air necessary to drive this process of conversion of juice into wine? And again, if you boil this and allow air to come in and out, that is still not sufficient to allow the formation of alcohol through the process of fermentation. So he thought there were microbes that were needed. And not only did he demonstrate as microbes, but there's a very specific type. So when he looked at this grape juice, he saw not all microorganisms look exactly the same. So what if you boil the grape juice, killing off all the microorganisms, and just add a single type of microorganism to the mix and see if you get the end result? And so he did this. He added the really small things that we now call bacteria. And then in a separate one, he added, still small, but not as small as bacteria, that are called yeast. And he looked at what the end result is. And what he found was really important, because bacteria did not yield the fermentation result that's desired. You get acidic products, which is not what you want in making good wine. But when you add yeast, you do get that fermentation into alcohol. And so what he demonstrated is, one, microbes are needed for fermentation, but also, two, it's a specific type of microorganism that's really important. And we now, of course, know that that's yeast. And so allowing those brewers and those people that are making wines to develop protocols to make sure that yeast is present and exclude bacteria was really important in those fermentation experiments. Once those kind of questions were addressed and the answers were starting to come in, and it was clear that microbes were present, you can then start to see how that would be an important finding to understand how disease could be caused. And so there was a variety of different beliefs, but one of the primary ones was that there are these vapors in the air that can make you sick, and then also imbalance of different fluids within the body. And so there were these different ways that people could be treated. But one thing that wasn't really looked at is the belief that microorganisms might be causing the problem. So there are two people that we want to focus on to really confirm that this is indeed the case, that there are microorganisms that can cause different diseases. So again, we have Louis Pasteur and we have Robert Koch. So these are the two individuals that we're going to talk about. There are, again, videos over here for each of them. I'm not going to show them. You can look at them. They're again, relatively short. But what we want to look at is Louis Pasteur. I have developed in quotes here. There were other individuals who proposed this germ theory. So he didn't come up with it on his own, but he really provided some really important evidence. So we're kind of calling him the major contributor to this germ theory, which states that microorganisms are responsible for disease. So obviously not all diseases, but there's a good number of diseases that we can say are infectious and are caused by microorganisms entering our body. And Robert Koch was also on board with this, and he was doing work in his own laboratory to actually support this germ theory of disease. And so we're going to look at Robert Koch's contribution. And so he came up with a series of guidelines, or kind of a procedure, that are called Koch's postulates, that allowed him to actually definitively link this microorganism causes this disease. And so Koch's postulates are 4 steps that need to be carried out to establish that connection. And so that's what we're going to look at. Skip those videos. And here is a picture of Koch's postulates. So I mentioned there are 4 steps, so there they are, and so we'll walk through them. In this case, if the goal is to say that microorganism A causes disease A, then when you look at that disease, so here you have disease individuals, in this case it's a mouse. Well, if you think there's a specific microorganism causing it, then you have to be able to isolate that suspected microorganism or germ from every case of the disease. So it can't be sufficient 9 times out of 10 you can say the microorganism is present. It's got to be present each and every time if it is indeed causing the disease. So that's step 1. Step 2 is you have to take that suspected microorganism and grow it. And so this is something that we'll study in more detail. They're called Petri dishes or Petri plates. And so on here you can see a diversity of microorganisms, but in each case the suspected microbe has to be present. And step 2 is actually being able to take that suspected microorganism. In this case, we're calling it this red bacterial colony here. So we aren't interested in all those other bacterial colonies, but that red one, if we can grow it in isolation, which means by itself, the term we use for that is pure culture. So the only thing growing on this plate now is the suspected germ, that microbe that we think causes disease. So we have to be able to grow it in pure culture. That's step 2. This is a little bit of an extension of number 1 here. If you read this, it says the germ is not typically found in healthy subjects. That's not always the case. There is these things that we call carriers that have the microbe in them, but don't always have signs and symptoms of disease. But most of the time, healthy individuals wouldn't harbor this potential germ. So healthy individuals typically don't have this suspected microbe. And what you're going to do in step 3 is take that pure culture of the suspected germ and somehow introduce it to this healthy individual, in this case through injection. And then you can follow that experimental individual, in this case the mouse, to see if it gets sick. Because if this microbe causes disease, you might indeed expect that an illness will develop. And not only that, you can then see and isolate that suspected germ from that sick individual. So you've kind of gone full circle from one sick individual, introduce it to a healthy one, they get sick, and then isolate that. And so that's what's known as Koch's postulates. So while Koch's postulates were tremendously important for, again, establishing that link, that germ theory of disease, one microbe causes a specific disease, to actually do this work in his laboratory, in Koch's laboratory, there were all sorts of techniques that we still use today. So I think it's number 3, lecture 3, we talk about staining techniques. But bacteria are really hard to see in the microscope, so being able to add color to them to see them is really important. Taking pictures of bacteria, actually allowing bacteria to grow. I mentioned Petri plates are really important. So how do you make Petri plates? It was in his laboratory that was kind of developed. So all sorts of contributions for Robert Koch. One of those stains is the Gram stain. And so Gram worked in Koch's lab and developed a technique that gave the end result of one of 2 colors. And so these are 2 different bacteria that have been put on a slide and then put through the same Gram stain procedure, but some of them retain a purple color, whereas others will at the end stain pink. And so this Gram stain is a really important, it's probably the most important stain within microbiology. And what we want to learn kind of pretty early on is what those 2 colors mean. And we're going to add to this throughout the semester, but we're going to see the Gram stain repeatedly. And so we can differentiate most bacteria into one of 2 groups. Gram-positive bacteria will stain purple. So we can say, oh, that's what a Gram-positive bacteria looks like after a Gram stain. And then Gram-negative bacteria stain pink. And so this pink color here would be representative of a Gram-negative bacteria. What makes bacteria stain differently is something we'll talk about in more detail later, and so I'll just give you a very short answer. It has to do with the structure of this important structure on the outside of a bacterial cell. We call it the cell wall or the cell envelope. And so those structures differ in these 2 types of bacteria. So Gram-positive have a different cell wall structure than do Gram-negative bacteria. And so that's all I'm going to say about it now, but we'll see that in more detail. We can now also talk about how we prevent disease. So these aren't necessarily in chronological order, but there are individuals who who contributed really important findings to how we prevent disease. And some of these were done even before we understood how diseases were transmitted. We hear about hand washing all the time in today's world, in this COVID world that we're living in. And so there's one individual that we can kind of trace back. His name is Semmelweis. That's his last name. He was a doctor in a hospital in Austria that when he took over the birthing clinics, he realized there's a fairly high rate of it was called childbirth fever. Another name for it is puerperal fever. But the moms and the kids were getting this at a pretty high rate, and it can have a fatality rate associated with it. So upwards of 20% of births were leading to death of mom or infant or both, which is pretty high. And so when Semmelweis looked at what was going on, he realized that those doctors were training medical students in the morning, and so they were working with cadavers. And then without washing their hands, they would go into the clinic where they would be delivering babies. And so he didn't have a term for it, but he called them cadaver particles. Maybe there are these things from cadavers that were being transmitted on the hands of these doctors and medical students. We now know these are, of course, microorganisms. And so he put a basin where they could hand wash, and lo and behold, that 20% mortality rate dropped down to below 1%. So we can trace Semmelweis back. His story is here if you care to watch for it. There's a little bit more sadness to it than what I gave you, but that's the gist of the story. Surgery is another important, obviously, another important field in medicine that we use, and there's amazing surgeries that can be done. The problem back in history was that surgeries often led to really bad wound infections, and so you probably had a 50% risk of developing a wound infection that may require amputation, or it could lead to death. So that's not a great outcome. So Lister is a surgeon who was the first to develop and put in practice using antiseptics to clean wounds and cleaning the incisions, and actually using these dressings that were soaked in a solution that would be an antiseptic as well. So we obviously know that having a really clean surgical suite is important in trying to prevent infections. And so Lister was the first to actually try this in practice in surgery, and did indeed decrease the amount of infections that he saw. And so here's a short story about him if you want to watch that. And then lastly, Florence Nightingale was an individual who took the cleanliness to not just the surgical suite, but really to the patient and the room and hospitals as a whole. So making sure that patients didn't sit in dirty clothes or dirty dressings or things like that, and making sure that beds and they were laundered between things and that. So that's obviously really important to reducing the risk of infection as well. And not only that, you probably may know that name, she founded a nursing school and put these practices into her training so that it was kind of disseminated across other nurses who were being trained through this practice. So again, I'm going to skip these videos, but feel free to watch them if you want to look a little bit more about each of those three stories. So we have several more. I think there's three more individuals. So John Snow is next. And he's considered the father of what we call epidemiology. And so in that same root is the same word as epidemics. And so what's really important in public health now is understanding, how disease is spread within the population, and actually trying to prevent that from occurring. So we obviously have a bunch of public health practices put in place today to try to prevent and control the spread of COVID-19. And so trying to prevent spread in a population was not done prior to John Snow. And so he used this practice of epidemiology, although it wasn't called that at the time, when he was studying an outbreak of cholera. And so cholera, we now know, is linked to water sources, and it leads to a diarrheal disease that can be very debilitating and lead to dehydration and death in a very short period of time. So a day or two, you can get so dehydrated that you can die. And so there is a bad outbreak of cholera in the city of London where he lived, not too far from his place. And so he started to go around and survey to see where the cases of this outbreak were actually clustered or located to. And this is the map that he actually used. So this is a map of this area of London. And these bars represent the number of cases at that address. And so you can see where all of these cases are clustered. In the dead center of them is this dot. It's a little hard to read, but it says pump. So this is known as the Broad Street Pump. And he just thought, well, if all of these people are going to have to get their water supply, they're probably going to go to the nearest location, which is this pump. There are other pumps scattered about. So here's a pump and here's another pump. So they're scattered throughout the city, but this Broad Street Pump obviously is going to be closest for these individuals. And so he actually studied this and came to the conclusion that it's this one water source, this well, that is supplying this Broad Street Pump that is leading to the disease. And so he was able to go to kind of the city council and get this pump shut down, and they actually indeed saw the number of cholera cases diminish, which was an important finding. So this video has a variety of different people. It has Pasteur in it, and it also has a little bit about Jon Snow, so you can watch that if you're interested in. Vaccines are hotly debated today. And obviously with the COVID-19 research and trying to get a vaccine present to help control this pandemic, it's a really important thing to understand. And so there's one individual that we trace a lot of vaccine biology to. There's obviously other individuals, but Edward Jenner is the individual that we want to focus on. And the disease that we're talking about is the disease smallpox. Cutting to the end of the story, smallpox is no longer on the planet because it was eradicated with a vaccination campaign. So it was the first disease to be eradicated from the planet because of a worldwide vaccination campaign in the 1970s. And so we can look back to how this vaccination program was even started. And if you want to watch this video, you can, but I'll keep it short. So there is a related disease to smallpox that's called cowpox. And it's called that because cows get it, and it can be transmitted to humans. And so those individuals, they are called milk maidens, that would collect the milk from cows frequently got this disease, cowpox. And it was much less severe, did not cause massive lesions across the body and lead to massive scarring and have a high mortality rate. And so milk maidens had this lesser form, but was also noticed is that they rarely, if ever, got smallpox. And so Edward Jenner hypothesized that their bodies were generating some protection from this initial cowpox disease that led them to be, he didn't use this term immune, but we now use this term immunology, and immune to this subsequent exposure of a similar disease called smallpox. So to test this is not an ethical experiment that could be carried out today, but this is how he did it. He took his gardener's son and exposed them to cowpox. So he got some of the pus from a cowpox lesion and then introduced it into this boy. He got sick with cowpox, developed a fever, got better, and he was fine. And then weeks later, he introduced him in the same way to smallpox. So he took some pus from a smallpox lesion, introduced it to the same boy. And luckily, his hypothesis was correct because he was protected and he never developed smallpox. And Jenner introduced smallpox multiple times to look at this protection. And every time, this boy never developed smallpox. So this was a really important finding, that we could introduce something into our body that led us to develop protection to something that could potentially kill us, like smallpox. Pasteur was working on something similar. So in his laboratory, he him and Koch were really kind of racing to establish what microbe causes what disease. And once those links were established, then Pasteur was also taking that known microbe that causes a disease and weakening it and seeing if that could be used as this vaccine principle. And so that's how many early vaccines were developed. Later in the semester, we'll talk about vaccine development, and there's a lot of different ways that it can be used today, but how vaccines can be developed. But attenuated or weakened strains was one of the original ways. That can still be true today, but there's so many others that we use as well. The last individual, another thing we talk about frequently with COVID is having treatments. So having some chemical agent that can be used for therapeutic purposes. And so Paul Ehrlich, we can kind of trace back to one of the early individuals who was looking for chemicals that could be used to treat these microbial infections. And so his goal was really to find one chemical that could be used to treat every infection. And so his goal was to find this thing called a magic bullet. It turns out he didn't do that. Because there's so many different microorganisms that we'll see in the next lecture. But his contribution is showing that chemicals can differentially affect these microorganisms. And so this was originally shown through staining, so that some bacteria will stain purple in a gram stain, some will stain pink. And so bacteria and microorganisms respond differently to chemicals. So what if we could find a chemical that negatively affected microorganisms that did not harm the patient, or the human in this case? So while he did not find a magic bullet that was able to be applied across all the microorganisms, he was able to find specific chemicals that were used to treat specific infection types. And so trypanosomes are microorganisms that we'll study pretty early on the lab side of things. So these are called protozoas, or in the class of protists. But they can cause diseases like Chagas disease and African sleeping sickness. And so these are still around today. And so Paul Ehrlich was the first to find a chemical that could be used to help treat them. Treponema pallidum is the bacteria, and it is the causative agent of syphilis. And so that's a sexually transmitted disease that can ravage the body if not treated and lead to blindness and mental deterioration and also death. And so Paul Ehrlich was able to find some chemicals that work for this as well. So this term chemotherapy we often think of for cancer, but if you think about what this means, it's chemicals, that's a chemo, used for therapeutic purposes. And so really any chemicals that are used to treat any disease could be classified as a chemotherapeutic agent. And so this is an important individual who established that link of chemicals being used to treat. And so now we have all sorts of antivirals and antifungals and antimicrobials like antibiotics that can be used to treat specific infection types. And so we can trace that to Paul Ehrlich. That brings us to the end of this first lecture. The next one that we're going to cover, so lecture 2, is going to focus on the different groups of microorganisms. So I know I used some terms like fungus and bacteria, and I threw out antivirals. And so obviously viruses are important. So the goal of the next lecture that you'll see is to understand those groups at just a really shallow understanding for the first pass. And then as we get deeper in the semester to actually dig deeper and understand those groups, at least some of them in more detail. So obviously viruses are really important to understand for human health, but so too are bacteria and also fungi. And so those are the groups that we'll kind of get more detail on. But first, we want to be able to know when we say microbe, what do we mean? And not only that, what are the basic characteristics of each of those groups? So until next time, that is the first lecture.